Okay, so today we have to use a spectrophotometer. Let's take this word apart. Spectrophotometer. I'm going to get class feedback on the recording. Meter, what does that mean to you?
Measure.
Measures, great. Photo, what does that mean?
Light.
So measures light. It's great. What's spectro mean?
Range.
A range. Outstanding. So, yep. So this thing will actually measure all the whole range of visible light, and it will measure the absorbance of that light. So if you pass a white light through the sample and it absorbs all the blue light, and you're measuring blue light, you'll see a negative reading on the absorbance. Okay? Everybody's an expert on spectrophotometry now. Remember that, make sure I'm getting this right though. We are doing absorbance today, right? Not emission. Okay. And so we're going to measure how much brown light gets absorbed. Or how brown it is, I think, actually. Yeah. Anyway, so we're going to select the wavelength, and the instrument is variable, so we have to designate which wavelength we're going to measure. And it will talk about what it will measure is the difference between what it sent to the sample and what made it through the sample. That's spectrophotometry. We'll have to make a baseline measurement, which means we're going to put everything that's the same except the actual reaction materials to see what type of light it normally absorbs, and then when that reaction is actually going, we'll see the real difference by being able to subtract the baseline. So, what are we measuring today? Enzyme activity. Can we get in there and just look at the protein working? No. So what we're measuring is a readout of a color change in the chemical as the reaction progresses. Okay? But part and parcel to our experiments is this is an enzyme. An enzyme is a type of what macromolecule? 1, 2, 3? Protein. So it's a subset. There are structural proteins and so forth, but enzymes are the ones that actually catalyze reactions. And the way they do that is they take on a specific structure facilitated by their actual amino acid sequence. So A, A, T, G, Y, W, R, Q, D, E, F tells the protein with the available chemical interactions within that string what shape to take on. Okay? So there are multiple forces acting on this structure. There's intramolecular, that is within the molecule, hydrogen bonding. There's actual covalent bonding between cysteine residues. I'm making a list here, so it's one of those things that shows up on tests. There is hydrophobic versus hydrophilic residues. So the hydrophilic ones, because in the cell you're in a water solution, the hydrophobic ones, the ones afraid of water, the nice fatty ones, tend to end up on the inside, insulated by hydrophilic residues on the outside. And I'm certain I'm forgetting one. I'll send you a note later. Yeah, that's how we get the shape from the amino acid sequence. It's not just a line of amino acids. It takes on a 3D shape. And that 3D shape is part and parcel to how it operates. It creates these very narrow spaces within it that are useful for recruiting the molecules into it. So there'll be like a little pocket on the outside. It can bind whatever the substrate's going to be. Substrate is the molecule that we're acting on. You need to add that to your vocabulary and use it constructively in our class. So they'll have pockets for binding that, and the shape can change of the enzyme itself. Typically changes shape as those different substrates are bound. So if it is catalyzing the joining of two substrates, let's call them A and B. A binds on one part of the enzyme, B binds on the other. The enzyme will actually change shape and bring those two together into an active site. And by forcing them together, typically that's how a reaction will occur. That is one example of a kind of reaction that's catalyzed by the structure of this. It could be, it doesn't have to move, but the point is the exact shape of the enzyme is what's going to let very specific molecules, I repeat that on purpose, to get into this structure and perform a very specific change. We're going to deal with one type of enzyme, peroxidase. So it's actually somewhat promiscuous in what kinds of substrates can actually get in there, so long as it has a peroxide in it, which can be a number of different things. Have another number of different things attached to it. All right, that's the overview. I have notes to do anything else specifically. This is it in cartoon form. Substrate will bind. It doesn't have to be two molecules being joined. It can be broken apart. It can be substituted. Anything that you're going to do in chemistry class, these things can do. They are catalysts. You'll notice that in the cartoon, we've drawn very specific shapes for these things to bind into the enzyme. It's like a lock and key mechanism. If it's shaped like an acetone, it can get into the active site. If it's shaped like an acetaldehyde, it will not. That smaller chemical group doesn't make it work. It's that specific. Once you have binding, the enzyme catalyzes reaction, you get the products. I'll get into this later, but since I'm a home brewer and we are doing a fermentation lab, and because the biology of it is interesting, I'll tell you that one of the major steps of brewing, and one of the things that we'll test in Chapter 6, is the activity of yeast on fermenting starches. And I'll give it away now. They don't ferment starch. What they have to do is use an enzyme to clip off little simple sugars one at a time. And that's actually one of the steps of brewing, is to make your starch into sugar before you can ferment it. Anybody that wants to know all the gory details of that wonderful process should come to not happy hour, but office hours, and have a conversation. You know, if you turn 21, you still remember me, I'll come talk to you about robust porters and everything. I'm just going to have you've seen this kind of scheme before about how scientific method works. We did that exercise with Mastermind to talk about reinterpreting our results, going back to the drawing board, starting with a hypothesis, and so forth. But look through this once, and we did that. And so specifically what we're doing today, we give guaiacol. Anybody know what guaiacol is? It's the roasty flavor in your coffee. So it's actually in bunches of stuff, generally a product of caramelization. And guaiacol. Plus we're giving it hydrogen peroxide. These will help us detect the activity of peroxidase. So peroxidase reduces peroxide to give off water as one of the products, and therefore the guaiacol becomes oxidized. So one thing is reduced, that's the water, or really, okay, let me do it straight. The peroxide here will become reduced. We're going to add hydrogens to this. Right now it's H2O2, it will become H4O2, and therefore split into H2Os. On the flip side of that, the yin to the yang is the guaiacol will become oxidized, and when it does that, it will have a change in physical properties. It will become brown, and that's all we're doing today is measuring the brownness. And so the reaction doesn't happen all at once if we're doing it right. It will progressively add more brown, so we're going to measure it over a time course, and hopefully that's what we'll plot. Today we're going to try to get to a working condition, baseline, and doubling the enzyme and halving the enzyme, and we'll take care of the others on the other